Alright, so today I'm going to talk to you about mutations. First of all, if you have not printed off the note sheet for mutations, you may want to do that now if you have a printer at home. If not, then you can just take notes on a piece of notebook paper, but you definitely want to take notes. I'm going to be doing just the first part of this PowerPoint, and I'll be talking to you specifically about gene mutations. Right after you watch this video and take your notes, you're going to want to watch the video that I'm going to do on how to identify different mutations, gene mutations, and so you'll watch that video next, and it should be in order in the folder. And I'll give you show you how to do the problems that go along with this. So, all right, so let's go ahead and get started. So first of all, you want to know what mutations are, and they are just changes in your DNA. So changes that occur in the DNA during replication. Remember we talked about replication last week, and it's the copying of the DNA. And you saw, the process of the DNA, the chains of nucleotides, splitting apart, then the new nucleotides coming in and pairing up. Well, in that process, sometimes there are mistakes that are made. Now, also, you have, with chromosomal mutations, sometimes the chromosomes don't separate like they should, and so there are mutations with that as well. But both of those have to do with DNA. So anytime there's a change in the DNA, that's a mutation. Now, a lot of times we think of mutations and we think of them as being bad, and many times they are. But there are some instances, which we'll talk about in our next unit when we talk about natural selection, that sometimes mutations can actually benefit organisms and help them to be better adapted to their environment. So, but that's in the future. So anyway, we're going to be talking about there's two major categories of mutations, and they are gene mutations, which is what I'm going to be discussing, and then chromosomal mutations, which Ms. Menachino will discuss in the other video. Now, gene mutations are going to be they're going to really affect just one specific protein, because genes are one section of the DNA that codes for a specific protein. So it's going to be, real specific what it's affecting. Whereas chromosomal mutations, a lot of times, are going to affect more DNA because this is where maybe a whole chromosome is missing or part of a chromosome is missing. And so a lot more DNA is affected with chromosomal mutations than with gene mutations. All right, so we'll go ahead and get started. Okay, so the definition for gene mutation, let's get that. And like I mentioned just a minute ago, it's going to be a change in the nucleotide sequence of a gene. So very, very specific. It's going to be affecting those specific bases, the adenine and the thymine and the cytosine and the guanine. One of the types of gene mutations that can occur is what we call a frame shift mutation. And to me, the name sort of kind of gives you an idea of what it's all about. Frame shift. So the reading frame is basically shifted over. And that happens because one or more nucleotides either gets deleted or inserted into the sequence. And so therefore, because those nucleotides are read 3 at a time, if you insert an extra base or you take one away, then it shifts the whole reading frame. And so this particular type of gene mutation, it can actually result in a completely different protein because the codon sequence is going to be altered so much. Okay, so that's a frame shift mutation. Now I have a little diagram I want you to look at, and then I'm going to do kind of a silly little example that might help you understand it. But let's go to our next slide here. All right, so here this is showing you what an insertion, that would be like if a nucleotide got added by mistake. So this is what it should be: AUG, CGA, UUA, UAC, and GGG. And you can see the amino acids, methionine, arginine, leucine, tyrosine, glycine, are the ones that should be made. All right, so then if you have the same sequence except an extra U gets inserted. Okay, so you see everything's the same until you get right here, and there's been an extra nucleotide that had the uracil base. It gets added. So now it still has to be read three letters at a time. So now it's going to be UUA, CGG, and then you got this G just hanging out here by itself. Okay, so that's a hint. When you see just one base or two at the end of a sequence, that tells you, okay, this is a frame shift. So if there's just one out there, then an extra nucleotide has been added. All right, now the other example, this is a deletion. That would be where a base or nucleotide gets deleted. So you see this is what it should be. Okay. And then we see right here you have a base that's been deleted. Okay, so here the C got deleted, but it still has to be read three letters at a time. So it just scoots over, and instead of UAC, it's going to scoot over as UAG, and then GGA, and then you end up with AA at the end. And so that tells you, okay, a nucleotide was deleted. But notice, in both cases, everything past that mistake, past that insertion or deletion, has changed. So it changes the amino acids after that change, and so that can cause the wrong protein to be made. Okay, so let me show you something real quick. This is just a silly little example that sometimes I do to help people understand it a little bit better. So let's say that I gave you a sentence, and the sentence was made up of nothing but three-letter words like the fat cat sat. Okay, so we all agree that's a sentence made of three-letter words. Now, if I came along and I said, okay, this letter, I'm going to delete this letter, but you still have to read it three letters at a time. So now it would be the fac, V-A-C, ats, A-T-S, A-T. See, you still have to read it three letters at a time. The fac, ats, at. Well, now the sentence doesn't make any sense. So you can see that it's making a completely different sentence. Okay, same thing would be true if we added an extra letter. Okay, so let's see if we can do that. So here, I'll do the same sentence. The fat cat sat. But this time I'm going to come in and I'm going to add an extra letter. But I tell you, you still have to read it three letters at a time. So the fat ka to sa, and then you got that T just hanging out there at the end. So you see that it completely changes that sentence. Okay, so that's kind of how you can think about a frame shift in terms of when you're talking about your sequence of nucleotides. If you insert an extra one or you delete one, it's going to completely change the amino acids that are produced. And so therefore, it's going to change the protein that's made. All right, so now let's look at the next type of gene mutation. And this is called a point mutation. And there's actually different types of point mutations. But a point mutation, and I want you to add this to your notes, is a substitution. So that's a word you'll want to remember. Substitution. So whenever you think of a point mutation, you should think of substitution. Okay, takes me a minute to write with this thing. Okay, so just like if we were at school and I was going to be out, I had to get a substitute. So that means that someone would come in my place, right? So it doesn't mean there wouldn't be a teacher at all. It doesn't mean there'd be two teachers. It means that there would be a teacher there in my place. Okay, so a point mutation that's basically what's happening. It's not that a nucleotide has been inserted or deleted, but instead it's just a change. So it involves a change in a single nucleotide. And so therefore it's going to affect just a specific codon. It's not going to, cause a bunch of different amino acids. It's going to change one codon, one amino acid. All right, so the first type of substitution or point mutation is a silent mutation. Now, if you were going to ask for a certain type of mutation, this would be the one to ask for because it's silent, which means that it doesn't cause any noticeable change. Basically, what happens with a silent mutation is you have a codon, UAC, is just one example. If we look this up on our chart, UAC is going to code for tyrosine. So I'm just going to write TYR under there. Well, if this C mutates to a U and now you have UAU, UAU also codes for tyrosine. So there's no difference. So if they code for the same amino acid, so I would write that down. Same amino acid. If they code for the same amino acid, then it's a silent mutation because it's not going to cause any change that would be noticed at all because that's not going to change your protein at all. So this would be the best type of mutation to have. All right, now the next type of substitution or point mutation is a nonsense mutation. And what happens in a nonsense mutation is it causes a stop codon early in the sequence. So basically, if it causes there to be a stop codon in the middle of your sequence, that's going to make your protein non-functional, and because it's going to stop adding the amino acids before the protein's made. And I always say that it makes no sense to have a stop codon early. So let's look at how that would work out. So if you had UAC, this was what the codon was supposed to be, UAC, and this nitrogen base here gets mutated to a G, so it went from cytosine to guanine, and now you have UAG. Okay, so there's your mutation. So UAC, remember, was tyrosine. But if it mutates to UAG, well, UAG is a stop codon. So if this happens, somewhere in your sequence early instead of at the end where it's supposed to, then that's what we call a nonsense mutation. And it's going to basically terminate the protein before it's completely made. All right, and then the last type of substitution or gene mutation is a missense mutation. And what happens here is the mutated codon is going to cause a completely different amino acid to be inserted. So if you had UAC, which we know is tyrosine, and that's U mutates to a C, and now you have CAC. Well, CAC is going to code for a completely different amino acid. It's going to code for histidine. Okay, so these two are completely different amino acids. So that's what we call a missense. Sickle cell anemia is caused by a missense mutation. So just that one amino acid being different or being the wrong amino acid can cause that protein in the blood cells to malfunction, and it the cells don't carry oxygen like they're supposed to. All right, so and they kind of get deflated and fold in on themselves. All right, so those are the three types of point mutations, which are all types of substitution. You have one letter that gets substituted with another letter. Sometimes it makes no difference at all, which would be a silent mutation because they both code for the same amino acid. Sometimes it causes a stop codon early, which is a nonsense mutation, and that's definitely going to lead to a non-functional protein. And then sometimes the protein is made, but the protein is not going to work properly because you have the wrong amino acid inserted. And so the mutation leads to a completely different amino acid. All right, so that is pretty much all that I am going to be talking to you about today. Tomorrow, or on your next video lesson after you do today's lesson, Miss Minakino will do the chromosomal mutations. Now, after you finish this, I want you to go in and watch the next video that I've done for you, and it's going to show you exactly how to identify those different types of gene mutations, and I'll show you how to work out the different problems. Okay, y'all have a great day.